Hi everyone, I'm Jan Witkowski at the Cold Spring Harbour Laboratory, and this is the occasion of the 89th Cold Spring Harbour Symposium on Quantitative Biology. And this year it's on senescence and ageing, and I'm very pleased to have Fred Rusty Gage from the Salk Institute with me. So Rusty, you were talking about line elements today. What are those?
Line elements are a class of molecules or DNA in our chromosome that have this strange property that they can move around in their DNA, and there's different types of them. There's something called transposons, and they're stretches of DNA that make proteins that clip them out, and they can then move to some other location in the genome. But so they're moving around. They call transposons. The other ones are called retrotransposons because they're a stretch of DNA, about 7 kilobases, and they can make an RNA copy of themselves. And then they have the machinery built into this that allows them to nick the DNA at some other location and insert themselves and reverse transcribe the RNA into DNA and make a new copy of themselves. So it's a copy and paste mechanism. And remarkably, cumulatively, all these what we call repeat elements constitute something like 40% of the total 3 billion DNA pieces that exist in every cell. So in normal cells, the protein coding sequences in the genome is 2% of the 3 billion pairs, whereas we're looking at 40%. Of those, they're not all active, but they're playing a role in what's going on.
Right. And what are the particular characteristics of the line 1 elements that you have been studying? The L1 elements? Yeah,
the line 1s are, as I said, these are these retrotransposons that copy and paste themselves in. And when I first discovered that they were active in neural stem cells and neural progenitor cells, was a bit of a surprise because we thought they were only in germ cells, but it turns out they're in the brain as well. And we studied for a long time just where they inserted, what activated them to insert in new locations, how frequently did it occur, was it region specific in the brain, a lot of the basic biology of it. Later we learned that most of this was happening when the cells were early stages of progenitor, we call progenitor cells, before they became neurons. But then we found out that later on, even as a mature cell, they don't make more of them, but the elements themselves leave the nucleus and reside within the cytoplasm. And in normal healthy conditions, they're sequestered in these features in the cell called condensates. But as we age and with disease, the condensates dissolve. And the RNA, which is sitting in there, gets converted into DNA. That DNA then hybridizes or binds to RNA, and the cell has an endogenous system, a surveillance system looking for foreign bacterial infected DNA, like a virus or something, and it's called the C-gas sting response. And that sees this floating around the cytoplasm, attacks it, and tries to kill it. And in the process of that, it induces a giant inflammatory response. And this is one of the features that is indicative of a neuron, healthy neuron, turning into what we call a senescent cell. It stops functioning, becomes inflamed, and toxic to its environment.
But you're studying these in, how do you study them in Alzheimer's disease? You can't do these experiments on humans, presumably.
Exactly. Well, so this is part of our experimental paradigm that we've developed over the years. We do want to look at human brain, living human brain. But of course, we can't just take a piece of brain out and study it. So we use a methodology where we take a skin biopsy from a patient who's been well characterized in terms of their cognitive capacity, maybe their genetic risk scores for the disease, how old they are. We have a lot of information about them. And we have a cohort of 150 young, adult, aged, healthy agers, and then we have this AD cohort. We also have a group of what we call mild cognitive impairment, which are on their way. And we take those skin biopsies, grow them in culture, and we have a method where we overexpress a certain set of genes, and it drives them directly into becoming a neuron. So within 3 weeks of overexpressing these genes, you can convert a fibroblast into a neuron. And it's a functioning neuron. You can patch clamp it with electrodes, you can, it has many features. But the important thing is, it retains the age of the cell from whom the biopsy was taken. And this is in contrast to what is traditionally thought of in these reprogrammed cells of iPS, or induced pluripotent cells, where you take the fibroblast and convert it into an embryonic stem cell, then make a neuron. If you did that, you'd lose the aging component of it. So now we've captured the aging component, and that's the cell that we have used.
The Alzheimer patients that you use, they are presumably, are they age matched to your controls? And presumably the Alzheimer's patients are all older.
Yeah, so the age matched controls and the AD controls are matched as much as we can. So for example, chronological age, they're between 70 and 90. Both the aged ones, so the controls are interesting because they're sort of a resilient population. These have no cognitive impairment. They're our baseline control. Interestingly, as a side note, we're now getting skin biopsy from people over 100 to really tap into that resilient question a little bit more.
And so you're looking at line elements in these normal controls on these age-matched patients. So what are you finding?
So I should say, just for clarification, we look at a lot of things. But what I focused on last night was on the surprising fact that the aging cells have a fraction of them that become, we call senescent. That means they stop functioning as a normal cell. They're not working well. They're not dying, but they're sort of resting in the state. But the key features, they're very inflammatory. They secrete all kinds of cytokines that excite their environment and disrupt things. And we found that there's this subset of Alzheimer's disease cells that are sort of penetrating this toxic environment throughout. And so we've wanted to find out what's causing that, what's causing this. And we, we had a hypothesis early on that somehow the line expression level was changed, that the amount of lines that were available had changed and there were more of them in this. And this is a sort of a global hypothesis. So we tested right away with drugs that had been used for AIDS. So one of the treatments for AIDS is to block reverse transcription. So the AIDS virus becomes active by being reverse transcribed and making it into a DNA integration into your genome. That's what makes it sick. And patients have been given these drugs for 20, 30 years.
Quite.
And of a few epidemiology studies, not large enough, but some have shown that people that take these drugs for 20 or so years have a less chance of getting Alzheimer's disease. So here's an epidemiological hint out there that this might be useful. So that the first study we did was to take these HIV drugs, put them on our cells, and we could dramatically decrease the inflammation that we saw.
Right, so these AD senescent cells, they have a shorter lifespan in vitro as well? Do they, or is it?
Well, we look at them generally at a month after we make the conversion, because they are already captured at that 1 month. They are the age that they were of the individual. Right. So we don't need to age them. They're already there. As soon as they become neurons, they are aged.
That's it. Yeah.
So that unlike other things where you have to sort of age them in culture for a longer period of time, since we have this direct conversion methodology, we don't need to do that.
So of course the next question has to be, these are all in vitro.
Yes.
So what happens, are there AD models of mice that can be used that have similar features?
Yeah, there are AD models of mice that we are doing this right now, of seeing whether or not there is a change. One of the things that we're particularly interested in is what is the mechanism by which the elevated amount of, or what is the mechanism by which the reverse transcriptase inhibitors, these AIDS drugs,
right,
how do they actually work? And we know that they block the ability of going from RNA to DNA. And if you do that, then you won't make the DNA that activates the C-gas sting response.
Right.
So we need to see whether or not that exact same process is going to work in the mice first, and we don't know that yet. We're in the middle of doing those experiments, but that's one thing that actually came up in the symposium today. We had a long discussion about what would be the best model for actually trying this out.
And I guess this question is somewhat related. So you've talked about lines in your induced AD cells, but if you look at other cells, other tissues in AD patients, do you find similar elevations of line?
So we have not done, so part of the problem is we've established this aged neuron, and we have to get an age equivalent astrocyte, microglia, parasite, and that's a whole other...
Well, I was thinking about other tissues like kidney and things.
Yeah, so we know for a fact that senescent fibroblasts, you can grow fibroblasts in a dish, and if you keep them growing for a very long time with many, many passages, they will eventually senesce. That means they stop dividing, and that's one of the definitions of senescence. Hayflick, exactly. Well, it turns out if you give 3TC or these drugs to the fibroblast, you can get them out of that senescent state through this gas thing. So yes, it does happen, at least in the skin and the brain.
So putting you on the spot,
yep.
When's the first clinical trial?
Well, it's interesting. There are now 3 companies that are designing these trials, and they've already started in some other diseases which have been identified as having line dysfunction. So phase 1, phase 2 trials are underway. AD trial hasn't done. People want to see that in these shorter trial studies, the way they want to see a signal. But I imagine I would be imagining in 2 years that there'll be at least 1, if not 2 trials underway.
So thinking about trials or human disorders, I was thinking that Gage find that first line insertion in factor 8.
Yeah.
Over 40 years ago now, in the early 80s.
It was in the early 80s, you're absolutely right. Yeah, Haig was a fabulous leader of this field. And he, whereas Barbara McClintock really, who studied here and worked here at Cold Spring Harbor, is the founder of the field, in fact, the absolute founder of the field. She discovered this, and for that she got the Nobel Prize in 1983. But Haig really took it from a molecular perspective to understand the mechanisms of how the life cycle of these lines work. And he has a whole, lineage of people. And I was very fortunate to have worked and enjoyed his company very much.
I was about to say you'll have to come back in 40 years and we'll see the progress. Well, if your drugs work, maybe you will be back in 40 years.
Yeah. Well, there you go.
Oh, thank you very much.
Is it okay?
That's
great.
fine.
All right, enjoyed it. Thanks very much, John.